Hi, my name is Nashielli and today I will be talking about iPS cells in regenerative medicine. Yamanaka is a Japanese scientist who thought, what if we could produce cells that could become any of the three germ layers? With this, we'd be able to replace damaged cells and tissues caused by various diseases, there'd be no fear of rejection, and could create various cells such as neurons, pancreatic, and other types of cells. Thanks, Yamanaka, for being the genius you are. Before discussing his findings, it's necessary to go over some terminology on cell potency. First, we have totipotent cells, which are like the queen bees and can even give rise to entire organisms, like my queen bee, Beyonce, who gave birth to 3 beautiful human beings. Then, these cells can differentiate into pluripotent cells, which can differentiate into any of the 3 germ layers, and these are going to be the ones that Yamanaka wanted to create. And finally, we have multipotent cells that make up the 3 germ layers and differentiate into specific tissue lineages. The first being the ectoderm, which include neurons and skin cells, the mesoderm constituting red blood cells and cardiac muscle cells, and finally the endoderm, which include lung and pancreatic cells. In Yamanaka's experiment titled The Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Cultures by Defined Factors published in 2007, he and his graduate student collected fibroblasts from mouse embryos and adult nude mice. Then these fibroblast cultures were treated, so to say reprogrammed, using transcription factors determined by Yamanaka in another series of experiments that could induce pluripotency. These factors were found to be OCT4, SOX2, c-MYC, and KLF4. So let's pause for a second. You know that saying that if it looks like a duck, and if it swims like a duck, and if it quacks like a duck, then it's a duck, right? Or maybe not in this case. Let's use this analogy to look at Yamanaka's experiment. He used this type of questioning to confirm that his induced pluripotent stem cells in fact closely resembled normal embryonic stem cells. He compared their morphology, gene expression of ES cell marker genes, and the way that they grow. So if Yamanaka could say that it looks like a duck, and that it swims like a duck, and that it quacks like a duck, then at this point he should have been able to say that it was a duck, right? In other words, that his iPS cells were somewhat equivalent to the pluripotent ES cells. Or exactly. It doesn't seem that this conclusion could be made just then. But the most important question is, does it act like a duck? Even though he proved that his iPS cells did indeed differentiate into the 3 different germ layers, scientists even today have struggled with making these cells act just like the specific desired cells. Yamanaka's work has contributed tremendously to the field of regenerative medicine and has been applied to various studies. Imagine a world where people like Selena Gomez and Steve Jobs wouldn't need to find someone else for an organ transplant. Or yet a world where people like my grandfather, who suffered from strokes, could be treated with stem cell therapies to regenerate damaged nerve tissue. Much work is yet to be done in this field, but the potential is great, thanks to Yamanaka's studies. Thanks for watching!